Hello everyone, welcome to the another lecture of genetic engineering and its recent advancement. Today we are going to learn about CRISPR-Cas system. Let's dive right in. Imagine you had a magical pencil that could rewrite the correct mistakes in a storybook. Well, in the world of biology, CRISPR is like that magic book, allowing scientists to edit the book of life itself. So, fasten your seat belt because by the end of this presentation, you will have a clear understanding of how this amazing technology works and why it's changing the way we look at genetics. Before we plunge to the captivative universe of CRISPR-Cas system, let's begin with the fundamentals. Have you ever experienced the desire to edit a photo in your smartphone? Oh yes? Then, well, imagine having the extraordinary ability to edit very blueprint of life itself. That's the essence of genome editing. Imagine our DNA as a colossal book, instruction manual for the constructing and operating living beings. Now, picture this book having a tiny typographical error, a genetic typo. Genome editing is akin to being the genetic detective which tracks down and corrects these tiny errors. For a long time, scientists have been on a quest to edit the genome, similar to what we might want to correct a misspelled word from the story. Genome editing is like a master architect of genetics, capable of making extravagant precise alteration to the DNA, much more like a skillful modification in intricate blueprints for constructing a building. Scientists have harbored this aspiration of genome editing for decades, but the path should be such precise, it should not be winding up in any form of uncertainty. Now let's unveil the stars of our show, the CRISPR-Cas system, one of the most astonishing discoveries in the modern biology. First, let's decode the acronym. CRISPR stands for the cluster regularly interspaced short palindromic repeats. It may sound like a tongue twister, but it represents a revolutionary system that was uncovered through years of relentless scientific pursuit. CRISPR-Cas, often dubbed genetic scissors, wasn't unraveled all at once. It is a culmination of undying dedication and curiosity-driven research. Imagine scientists and detectives trying to solve a complex puzzle with a vital, precise missing blocks that changes how they are faced. Now, discovery of CRISPR-Cas is credited to brilliant work of two exceptional scientists, Jennifer Doudna and Emanuela Charpentier. In a historic moment of scientific achievement, they jointly unveiled the intricacies of this genetic marvel. In recognition to their pioneering contribution to the science, Jennifer Doudna and Emanuela were honored with the Nobel Prize in Chemistry in the year 2020. This prestigious award underscored the profound impact of their work, which has opened the horizons for precise genome editing, transforming our capabilities to modify and understand the genetic information. But why? This CRISPR-Cas technology is so vital. Now, imagine having the power to control the genetic diseases with the same ease as proofreading a document for typos. Envision cultivation of crops that can withstand the pest and thrive in face of drought. Now, guaranteeing a stable food supply in our growing population. This is an inspiring change in case of CRISPR-Cas technology. It's nothing short of a genetic revolution. So today we embark on a voyage to discovery, exploring the depth of this remarkable technology and uncovering why it has sparked the waves of excitement around the realm of science and medicine. Now, let's journey back in time to uncover the intricate story behind the discovery of CRISPR-Cas system. Imagine this as voyage of scientific exploration akin to the peeling back of the layers of hidden treasures chest. The CRISPR-Cas system, as remarkable as today, it didn't unveil itself in a single moment of revelation. Rather, it's a product of relentless scientific inquiry, curiosity, and collaboration. Picture this challenge akin to solve as elaborate puzzles, which crucially pieces together the missing pieces. In early days, the keen-eye scientists observed something extraordinary within the DNA of bacteria. Now, they are cryptic repeated patterns. These patterns, like a series of mysterious symbols etched into a genetic code, caught their attention. It was as if they had stumbled upon a secret language written within the very genes of these tiny organisms. However, the full significance of these repeating patterns didn't come apparent unless and until it was 2000s. Researchers from around the world began to connect the dots, gradually unraveling the cryptic message encoded within the DNA. It was like experiencing a cascade of eureka moment that fundamentally altered our understanding of genetics. Imagine these eureka moments as fitting together the pieces of immense intricate jigsaw puzzle. Each revelation, no matter how small, brought us closer to the comprehending the grand picture. The journey involved brilliant minds collaborating and sharing their insights, much like an assembly of collecting masterpieces. Today, we embark on the exploration of these remarkable milestones in the CRISPR research, each stepping a stone that led us the inspiring technology as we have our disposal today. So, let's begin our learning. Now, let's dive straight into the heart of our topic, the fascinating CRISPR-Cas system. Now, imagine as this moment you open a user manual of a captivating new gadget full of surprises and possibilities. First, let's decode the mystery of CRISPR. CRISPR is a nature's indigenous immune system for a bacteria. Just as we human beings have immune system to defend ourselves from infections, bacteria rely on CRISPR to defend themselves against the viruses like bacteriophage. CRISPR isn't one size fit all the solution. There is a various type of CRISPR system akin to different superhero team, each armed with a unique superpowers for a particular job. But why should we be so fascinated by these Cas protein? Well, think of them as scissors and glues of the genome editing world. They are remarkable tools that rolls their sleeves and perform the intricate DNA edits. In a nutshell, CRISPR is nature's indigenous way to help bacteria defend themselves from the viral invaders, and Cas protein are those precise instruments we have harnessed to carry out the art of genome editing. It's a bit like having a Swiss army knife, but for the world of genetic engineering. So hold on to your seats as we journey deeper into the remarkable world of CRISPR-Cas. It's a universe filled with endless possibilities. Today, we are your guide to this thrilling exploration. Now, let's take a closer look at the parts that make up the CRISPR-Cas system. Think of it as understanding the key component of your favorite video game or puzzle. First up, we have CRISPR arrays. Imagine these as storage unit where the CRISPR keeps its memory of past encounters with different viruses. It's like a library that stores information about bad guys. Next, we have got Cas protein, and there is a various type like Cas9, Cas12, Cas13. These are like the action heroes of the CRISPR story. They are ones that carry out the precise editing of DNA, just like a surgeon with a scalpel. Now, you might be wondering, with the PAM equation, PAM stand for the protospacer adjacent motif. These are like a landing pads This Cas protein needs to recognize before they can start the editing of DNA. Think of it as a secret code that allows to find their targets. So to sum up, the Casper array stores the viral information, Cas protein are the editors, PAM sequence are the GPS coordinates that helps Cas protein to find the right spot for editing. Understanding these components is a key to unlock the magic of CRISPR-Cas. As we have seen how all of these work together, we can go ahead. Now that we have met the CRISPR components, it's time to see how they work together in action. Imagine this as a step-by-step recipe for making your favorite dishes. First, let me walk you through the basics of CRISPR genome editing. It's like giving our superheroes Cas protein a mission, finding the specific piece of DNA that needs to be edited or changed. The Casper can target both DNA and RNA, but how does it know which one to go after? Well, think of it as having two different languages in a book. CRISPR can read both, so it can target and edit the right part. The key is specificity. The CRISPR can find the sentence it needs to change in the entire book of genetic codes. It's like finding a needle in a haystack. So, in simple terms, the CRISPR is like a genetic detective that can find and modify a specific DNA as well as RNA sequences, helping us to correct the genetic spelling mistakes, or what we call it as typos. We will explore this process step by step, so don't worry if it seems like a big magic right now. By the end you will see how it's completely fit together. Now let me talk about the mechanism in much detailed version. So let's dive deeper into the mechanism of CRISPR genome editing. Imagine this as a detective CRISPR that successfully and carefully searches for clue in editing a genetic code. First, CRISPR need to recognize the target, as I have already stated, which can be either a DNA or a RNA. It does this by matching a small piece of genetic code called as CRRNA with the target's genetic code. Think of it as finding a match puzzle pieces. When CRISPR finds the right matches, it knows exactly where its right place is. Now let's talk about another step, that is formation of CRISPR RNA complex. Now let's begin our studies. So here in this step, our detective notebook filled with information about the target, pieces of genetic code that matches the target. To make a CRRNA, the CRISPR uses its memory stored in a CRISPR array. It's like creating a personalized map for each mission. Once cRDNA is ready, CRISPR is armed with the information it needs to edit the target DNA or RNA. Now another important part is trans-RNA. But wait, the CRISPR often has a pattern in crime fighting. The trans-RNA, or like a transfer RNA, is like a trusty sidekick that helps CRISPR do its job. It helps guide CRISPR and Cas protein to the right spot. It's like a GPS that ensure they have reached to their right location. Now when cRDNA and trans-RNA join forces, they form a powerful team. It's like our detectives and sidekick teaming up together to tackle a particular tough case. Together they help CRISPR locate and bind their target RNA or DNA, ensuring that Cas protein will edit the right genetic code. Now we have found the targets. It's time for action heroes to perform the editing part. So when CRISPR and Cas protein meet the target, it's like a superhero responding to distress call. They activate and get ready to make the changes. So after this step, finally the most critical step is DNA cleavage. The Cas protein acts like a molecular scissors, cutting the DNA at precise spot specified by cRDNA, specified by cRRNA. It's like as if our superheroes has disarmed the villain, preventing it from causing any further more harm. This cleavage triggers the cell's repair machinery to fix the DNA, allowing us to either correct the genetic codes or genetic errors or introduce a desired changes. Yes, it is also possible. We will see it shortly. So, in a nutshell, the CRISPR-Cas system is a precise genetic editing tool that can find targets and cuts DNA or RNA thanks to the detective work and the superhero, the Cas protein. Now, the Cas9 and double strand breaks, we will study these in a much detailed form now. So, let us focus on our superstar Cas protein pin within the CRISPR-Cas system, Cas9. Yes, the protein name is Cas9. Think of Cas9 as a superhero that can cut DNA with incredible precision. But how does the Cas9 do it? How does it perform the cutting function? Imagine Cas9 as a pair of molecular scissors. It has a built-in GPS. As I have explained earlier, it's cRNA or transfer RNA that guide it to the exact spot of DNA where it need to be cut. Once it arrived at the target, Cas9 makes a precise cut at that particular location, just like a chef slicing few vegetables. But here's the interesting part, Cas9 doesn't just make one cut, it actually creates a double stranded break in DNA. Think of it as cutting both sides of a zipper. This doubles the standard break that triggers the cell's repair machinery, which can harness for a various genetic editing purposes. Now, why this double stranded break is so important in the genome editing? Let's see. Let's consider an example. Imagine you have a sentence with a spelling mistake in a book. Cas9 is like editor which cut out the wrong letters, creating a particular gap. When cells tries to fix this gap, it can either repair it exactly as it was earlier, allowing us to correct the errors, or it can introduce a new genetic material like adding new sentence to the story. This is how Cas9 helps us editing these genes. These implications are enormous, trust me. We can fix genetic diseases by correcting these mistakes or enhancing a particular trait within a crop to make them resistant for pests or diseases. So again, Cas9 is like a precise scissors of CRISPR's world. It introduces a double stranded breaks, opening the doors to the world of possibilities in the genome editing part. Now that we know how the Cas9 is a superstar in the CRISPR world, let's see how it compares to the follow the Cas protein, Cas12 and Cas13. Cas9, Cas12 and Cas13 are like three different tools in a genetic toolkit, each with its unique abilities. Let's break down their differences and similarities. Cas9, as we can see in this particular picture, Cas9, our precision scissors is excellent at making a specific double stranded breaks within the DNA. It's like having a surgeon who can precisely cut a tumor without harming the surrounding tissues. On the other hand, the Cas12 and Cas13s are like unique talents. The Cas12 can target DNA just like Cas9 has an added bonus. It can also target RNA. Yes, you hear it right. So it's like having a multi-tool that can cut both papers and fabric. Now, what are Cas13? Cas13 is specializes in RNA cutting. Is like a surgeon who can work on RNA molecules which are essential for protein production. Cas13 can cut RNA molecules and in some cases even control genetic expressions. So, what are some real world application of these Cas protein? Let's look it as in a closer look. Cas9 have been used to correct the genetic diseases. It's real now, my friends. It's like editing the genes responsible for sickle cell anemia. It's like fixing a broken car engine, making it run very smoothly again. The CRISPR-Cas, the Cas12 has found its places in diagnostic. It can detect the specific DNA or RNA sequences within the virus or bacteria. Imagine it's like super sensitive alarm system that can detect the intruders. Cas13 with its RNA targeting ability is being explored for potential treatment of diseases caused by RNA virus, particularly flus. It's like having a key to turn off the engine of a runway car. These Cas proteins opens door for various genetic editing and diagnostic applications, each within its unique set of tools for specific job. Now that we have understood how CRISPR-Cas works, let's talk about how these actually gets into a cell. Think of it as delivering a package to a specific address. Now there are several methods for delivering CRISPR-Cas components into a cell, and each method is like a different way to send that particular package. Let's explore them one by one. One common delivery method is using viruses. These tiny delivery trucks can carry CRISPR-Cas components into a cell. It's like mailing a particular letter inside an envelope. Now, another approach is electroporation. Imagine using a tiny electric shock to create temporary opening within particular bacteria, allowing the bacterial membrane to create these pores, and CRISPR-Cas component can slip inside the cells like a guest at a party. Sometimes we can also use nanoparticles, which are like magic carriers that can transport CRISPR-Cas components to a cell. It's like a sparkling fairy dust to making cells more receptive. Now let's talk about the two main categories, the viral and the non-viral deliveries. Viral deliveries is like enlisting a professional courier service. Viruses are very efficient in getting inside a cell, but there can be safety concerns and the immune response to be considered. On the other hand, non-viral deliveries is like sending package through regular mails. It's safer, but it may not be as efficient in some cases. Both methods have their pros and cons. Scientists choose their best approach based upon a specific need of their experiment or treatment. For example, in gene therapy, where we aim to treat genetic diseases, scientists might choose viral delivery because of its efficiency. It's like using the faster delivery services or urgent packages. But in some research setting, like studying a gene function in the lab scenario, non-viral methods might be preferred because they are safer and less likely to cause any unwanted side effects. Now let's tackle an important topic in CRISPR-Cas technology, that is off-target effect. Think of it as unintended consequences where we try to make a precise understanding off target effect. So, off target effect occurs when CRISPR-Cas doesn't hit the exact spot where it is supposed to be. Imagine that you are trying to fix a typo in a book, but you accidentally erase a very nearby word that was very necessary for the complete meaning of that particular sentence. These off target changes can lead to unexpected outcomes and potentially cause harms. Just like making the wrong edit in a computer program can crash the entire system. So, fortunately, scientists have come up with the strategies to minimize these off target effects. It's like adding safety nets where you work on a tighter rope. One strategy is using modified Cas protein that are more precise, like using a laser guided tool instead of regular ones. This helps to ensure the CRISPR-Cas only edit the intended DNA or RNA. Another approach is designing better guide RNA. Think of these as a GPS coordinates. By improving the accuracy of these coordinates, we can reduce the chances of going off the target. Now, importance of specificity. Why the specificity is so crucial in case of CRISPR-Cas editing. Imagine you are an artist working on a masterpiece. You need to paint precisely in a right spot to create a beautiful picture. In genetics, precision is essential to avoid the unintended consequences. We want to make sure we are editing the right genes without affecting any nearby genes or causing any further problems. For example, in treating genetic diseases, we want to correct the disease causing mutations without introducing a new mutation. It's like fixing a leakage in a pipe without causing more leakage. By improving the specificity of CRISPR-Cas, we can make genome editing safer and more reliable, just like aiming for a bullseye in a dart game. Now, let's fast forward to present and explore some cutting edge developments in case of CRISPR technology. Think of these targets as upgrades to the already available amazing tools. Cutting edge developments in CRISPR technology. Scientists are constantly pushing the boundaries of CRISPR, and one of the exciting recent advances in the development of even more precise and versatile tools. Imagine upgrading your smartphone to the target models with improved features. Similarly, scientists are upgrading CRISPRs to make it even better editing the genes. Prime editing and base editing. Two game changing technologies in CRISPRs are prime editing and the base editing. Let's read about them. Prime editing is like a word processor for DNA. It allows scientists to rewrite the genetic code with extreme precision, inserting or deleting specific sequences without causing double strand breaks. Is like editing a sentence in a book without ripping off any particular pages. Now, what are base editing? Base editing, on the other hand, is like a genetic spell checker. It enables scientists to convert one DNA base into another DNA. Correcting these genetic mutations are like typos in this particular word, is like as if you could change a cat to cot in a text document. These technologies are incredibly exciting because they expand the possibilities of what we can do with a CRISPR. We are like adding new tools in a toolbox, making genetic editing more accessible and precise. For example, the prime editing is being used to correct genetic diseases with high accuracy, and base editing is helping us target a specific mutation associated with any kinds of diseases, just like sickle cell anemia. These advancements are like turning the CRISPR tool into a Swiss army knife for genetic engineers, allowing us to tackle a wide range of genetic issues. So, while we have learned all these technologies, you guys must be thinking, so if these enough tools are available, what is the current research scenario? Currently, a lot of labs within India as well as within the abroads are using these technologies for their genome editing part. They want to study the function of each protein or each particular genes. So, for this purpose, one of the widely used methods that you have already learned in previous lectures, just like gene knockout or gene particularly introducing a new genetic or new genes. So, for all these purposes, these CRISPR-Cas system is widely studied. So, a particular gene 1 can be replaced with gene 2 and we can introduce this particular entire editing in the genome of particular bacteria, viruses or anything else, and now we can see what is the effect of these genetic changes by looking at the physiology of those particular bacteria, viruses or even these labs are taking this CRISPR-Cas system, optimizing for their particular organisms and looking at the after effects. Now, you must be thinking, if all of these are possible, is there any ethical consideration? So, as we venture further into the world of CRISPR, it is essential to address the ethical concerns that comes with this powerful technology. Imagine this as a moral compass guiding us through the uncharted territory. Ethical concerns surrounding CRISPR-Cas is a potential to edit genes opens up the ethical dilemma. One major concern is the unintended consequences of genetic editing. For example, when we edit one genes to cure a disease, we might inadvertently affect the other aspect of a person's health. Now, think of it as a delicate balance act. We want to heal, not to harm, and to ensure the safety of CRISPR treatment is at the top priority. The another concern is the possibility of designer's baby. So, CRISPR could theoretically be used to select a certain traits in an unborn child, such as eye color or a particular height. So, this raises a question about what should we do and what shouldn't be modified in the human genome. So, there are different examples in the world where people have fictionally created babies having the ability for a different eye color or maybe different fingers. Maybe people have even created comic character having 4 hands. It's like in Avatar, as I can say, in a Hindu religion, like a Vishnu avatar. So, imagine your child having 4 arms. So, is it the right way? So, no, it is like a game changing technology, but it have its own limitation. If these limitations are not monitored properly, people might end up having a lot of variations within a human system or within any bacterial or virus system. Now, imagine you have created a particular virus that can actually treat human beings, but imagine these viruses do not have the accessibility or a particular trait. You have mistreated this particular gene. Now, contrarily, it has become a very deadly viruses that can have potential to kill a lot of humans, animals and other organisms. So, it is like we have to take very precise step and very profound steps for these using of these technologies. In further subsequent lectures, we will study more about this ethical consideration and a detailed mechanism of editing a live particular gene. So stay together, we will learn all these in our next lecture. Thank you very much. Happy learning. I will see you in the next class.